Okay, hi guys. Welcome to our session on the nervous system. What are we talking about today? We're going to do an overview of the nervous system and its various divisions very quickly. We're going to do a really quick review of neurons and action potential, how they work, linking that into reflex arcs and what a reflex arc is. We're going to look at muscle cell and nerve interactions, how they work together. And then finally, just very briefly introducing you to the idea that drugs impact synapse interactions between neurons and diseases of the nervous system. So the nervous system obtains sensory information. It processes sensory information and sends commands to effector cells. Now, those may be muscles, they may be glands around the body, effectively saying, okay, this is the information, this is what we're going to do with it. The key thing when you're studying the nervous system is not to be anxious in any way about the topic. It's really not too much that you need to know about it. So we get sensory input from our environment, our eyes, our mouth, all our different sensory input mechanisms, our ears, etc., detect our environment around us. Remember, that's one of the Mrs. Higo characteristics, is sensing the environment around us, sensitivity to our environment. That information is then processed by the central nervous system, including the brain, spinal cord, and peripheral nervous system, and then sent back out via the peripheral nervous system for motor output to effector cells, saying, this is what we're going to do with the information that we've been given. And that might be something as simple as contracting a single muscle, or as complicated as coordinating many different muscles in order to respond to in the correct way to the stimulus. If you're a very basic organism and you're watching this video, congratulations. However, it would be surprising because if you have radial symmetry, such as a hydra does, they only have a nerve net. So this is a way of processing information, but it's a very basic processing of information. It's not, they're not able to process high-level information and carry out complex tasks as a result. Complex responses, really, I guess is maybe. But what we see in bilaterally symmetrical organisms, that means organisms that you could draw a line of symmetry down the middle, including humans and most animals that you can think of, is cephalization. This is a really important concept. Cephalization is the aggregation, the congregation of the nervous system towards the front of the organism. Okay, this is because that's where most of our sensory input needs to come from. So we want to minimize that distance. So in humans, of course, our brain is in our head, and that enables us to have very quick impulse, very quick messages between our eyes and our ears. The head is a sensible place to have this data processing, because that's going to be closest to the region receiving information about the area that you're heading into. Okay, if it's at your back end and you move away from your brain, then there's a slight delay. And remember, every little optimization of the system counts when it comes to evolution. So the nervous system in most bilaterally symmetrical organisms, including humans, consists of two major branches. You've got the central nervous system, the brain and spinal cord in vertebrates, and then you have the peripheral nervous system, which is basically everything other than your central nervous system. That includes the nerve fibers themselves. Remember, a nerve fiber is made up of multiple neurons. Those are the nerve cells. And the ganglia, which are the clusters, the gangs, if you like, of cell bodies of the neurons. They group together in little areas. Remember, a neuron is a really long structure, so that cell body is just a tiny part of it. So if we look at the major branches of the peripheral nervous system, so central nervous system, very straightforward, is brain and spinal cord. That's where most of the big data processing takes place. The peripheral nervous system that comes around that that goes out to the rest of the body. It has various branches. You have the somatic nervous system. Somatic nervous system is when you think about controlling a part of your body, such as the muscles in your leg, your arm muscles, or anything like that. Those that's the somatic, because that's cross. You choose your, you've got some of them on, some of them off. It's the somatic nervous system. The autonomic nervous system, as its name implies, is more automatic. These are nervous impulses that you're sending out, whether you want to or not. You have a degree of control over some of them, but not much. So the autonomic nervous system can be split into three divisions. You've got the sympathetic and parasympathetic divisions. You can think of the sympathetic division as the fight or flight division. These are the sympathetic division basically speeds everything up, gets you into kind of ready for action. The parasympathetic is sometimes referred to as feed and breed. It helps you, it switches on those things, it's more relaxed. It slows down heart rate, slows down breathing rate, and that's controlled by the parasympathetic division of the autonomic nervous system. There is one other division called the enteric division, occasionally referred to as the second brain, because the enteric division is the most autonomous division of the nervous system outside of the central nervous system. It can actually act without the central nervous system. And the enteric division is basically in charge of your digestive system. So, and food entering enteric, your body. I've often thought that my stomach is a second brain, and it turns out that in a way that's slightly true. Reflex arcs. So a lot of our responses to stimuli are reflexes. We don't actually have to think about what we're doing in order to manage these particular tasks. You have sensory neurons that come from sensory receptors. So sensory receptors are part of your sense organs that detect changes in stimuli. So for example, you have sensory receptors in your eye, you have sensory receptors in your skin. So all kinds of different types that detect different stimuli, be it vibration, be it heat, be it light, etc. These send nervous impulses to the central nervous system along what are called sensory neurons. Okay, so sensory receptors to sensory neurons. That goes to the central nervous system. In the central nervous system, there are interneurons, also sometimes called relay neurons, that process this information and send it out to the correct motor neurons. Now think about life. Motors make things happen. A motor neuron takes the signal to the effector cell that is going to affect a change. So an example of this is when a doctor comes along and randomly just, hits you in the knee. This stretches a ligament that causes sensory receptors in your legs to send a nervous impulse up your leg by the sensory neuron to the spinal cord. Now this doesn't even need to go to the brain, which I think is amazing. The interneuron in the central nervous system responds to that message and sends out automatically a reflex down the motor neuron. Remember that motor neuron is going back to the effector, which in this case is the flexor muscles, causing your muscle to contract and kick the doctor. Okay. Now this is not an evolutionary development for protecting you against healthcare physicians. This is actually interesting. A lot of people don't realize this, but this stretching of the ligament is an indication that your body is going to fall over. And so contracting the muscles enables you to stay standing up. It's actually a reflex that helps you to stand. There's all kinds of other reflexes. For example, a really nice one to do is to look at, you can't look at it yourself, but you can look at it in a neighbor, is to have them be in the dark for a while and then shine a bright light, not too bright, in their eyes. And you can see the reflex of the contraction of the iris, their eye. Now that's automatic. We don't have to think, oh gosh, I've got too much light going into my eyes. It's going to be really bad for my rods and cones in my eyes. No, your body does it automatically. So reflexes are very fast. Think about the fact that you do this automatically, and it's survival. It's a survival reflex. It helps you to survive in some way. So you don't need your central nervous system to process the information on a higher level. It can just instantly send it out. So just a quick recap. Remember that your nerve cells have these very odd shapes with the cell body at one end and then this very long axon, this strand coming off them. The strand wrapped in other cells called Schwann cells, which are made of myelin. That's going to be important in a moment. Just want to give you a very brief overview of the of the action potential as it moves down a neuron. This is really important, and it's something that could well come up on the AP. Remember, the resting potential of a neuron is maintained by sodium-potassium pumps. Sodium-potassium pumps are really useful things to know about, not only because they're very important in your body, but because they're really nice examples of two things. They're really nice examples of co-transport, that is two molecules being transported together, two or more molecules being transported together, remember. And they're good examples of active transport. They require ATP. Okay, this is a big job for ATP in your body, is maintaining the sodium-potassium pumps. So, in nerve cells and neurons, sodium is pumped out, potassium is pumped in. It's actually a 3:2 ratio of these two. And that helps to maintain this resting potential, which remember is negative. The inside of the neuron is negative compared to its outside. When the stimulus, which is the depolarization of the neuron, happens, some sodium channels are opened, okay? And if it reaches the threshold, remember this is an all or nothing response. If it reaches the threshold level, an action potential is triggered. More sodium channels are open, and the potassium channels at this point are closed. This causes the interior of the cell to become positive, and you can see on our membrane potential chart, the charge shoots up, becomes more positive. This is the action potential. Then, once the inside has become positive, these gated channels close, okay, and become inactive. So the sodium can no longer flow into the neuron. As that is happening, then potassium channels open and potassium rushes out, moving away from the positive charge on the inside out to the negative charge on the outside. This causes a repolarization, and always it'll overshoot slightly. Potassium channels then close, causing a brief undershoot, and then it's returned to its resting state with the sodium-potassium pumps pumping it back. So the key thing to remember, sodium-potassium pumps are maintaining this gradient. Negative on the inside, positive on the outside. First thing that happens, the sodium gates open, sodium floods in, depolarizes. Then the potassium gates open and potassium flows out, repolarization, and then we get returning to the resting state. The depolarization of one section of the axon causes the depolarization of the next section, and so our action potential moves along the neuron. Now this is sped up by these Schwann cells between what I love, one of my favorite names in biology, the nodes of Ranvier, okay, or Ranvier. These nodes allow the neuron impulse to jump between these across the Schwann cells, and it actually speeds up nervous transmission. The Schwann cells also act as an insulation, preventing one neuron from accidentally causing a depolarization in a different neuron, which shouldn't be depolarized. Of course, when it reaches the end, as you know, it reaches a synaptic cleft, okay, the synapse between two neurons. And this is so important. It's often, you may not realize it, but this is really, really critical for controlling your nervous system. It's amazing to think that all the memories you have, all the deep thoughts you have, if you have any of those, they all, and the shallow thoughts, they all are controlled by the junctions between one neuron and the next, and how those neurons connect to one another. So it's crazy if you think back to your favorite moment in the last week, or your last birthday, or a memory, that those things are controlled by your synapses. It's bizarre, it's strange in a way that these tiny gaps between neurons basically are acting in this way. It's like a computer. It's computing power. Which neurons connect to which other ones, and how does that work? Remember, when the action potential arrives at the synapse, this causes vesicles containing a neurotransmitter to be secreted into the synaptic cleft. They diffuse across the synaptic cleft. You've got to remember, this cleft is very, 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 very narrow, so the diffusion doesn't take a very long time. They activate the neurotransmitter, they activate the receptors on the receiving neuron, and that in turn will cause depolarization or some other response from the receiving cell. And I say so, or some other response, because of course that other cell could be something like a muscle. Okay, when nerve cells reach muscle cells, the acetylcholine, that neurotransmitter, diffuses across this, the gap, the neuromuscular junction, to the muscle cells. And within those muscle cells, we get one of those cell signal pathways that we've discussed before. In muscle cells, this is calcium that flows in and binds to actin. Okay. Now the actin is then bound to by myosin, which acts like a set of rowers, pulling the actin past it. Don't worry about the other terms in that one, but just understand you've got your actin, which is acted upon, and the myosin, which pulls the actin together. Now, obviously, one of these fibers on its own, not particularly strong, but working together, thousands and thousands of these, they can control the contraction of a single muscle, and so the movement of your body. I already mentioned that these junctions act like a computing system. Many of the synapses can be either inhibitory or excitatory, and so these interact together to determine whether a neuron is going to fire and pass on a message or whether it's not going to fire. And the interaction of all of these different synapses comes to that kind of conclusion as to what's going to happen. So, lots of different molecules can impact synaptic transmissions of messages. They can either inhibit the neurotransmitters, they can mimic neurotransmitters, they can do all kinds of different things. They can cause more neurotransmitters to be released than otherwise would be. They can stop inhibition, they can promote inhibition. And these things create depressants, stimulants, hallucinogenic drugs. All of these different drugs are acting on the synapses. So one of the things that you should definitely do is a little bit of research about how drugs impact synapses. There's a great website, Learn Genetics Utah, and it's called Mouse Party. What you need to do is to select each mouse and research how is that particular drug impacting a neurological synapse. Okay. So that's one of the tasks that you need to complete. Research at least 2 or 3 drugs for that one, please. And then the second thing I'd like you to do is to research how the nervous system is impacted by disease. The one I'd like everybody to look at is multiple sclerosis, and then you can either research Parkinson's disease or Huntington's chorea to work out how do these diseases impact the nervous system, what causes them, and what treatments do we have, if any. Try to relate your research back to how neurons function and how synapses function. Okay, good luck with your research.